So now we're going to talk about enzymes. Not all of this is in your book. In fact, there's only a little bitty paragraph in your book about enzymes, but the EOC and the test makers like to focus on enzymes. So here we go. Proteins that act as a catalyst, lowering the activation energy and increasing the rate of chemical reactions. Now, there's a couple of facts about enzymes. They're specific to only one or two chemical reactions, most often only one. The enzyme's name is usually from the chemicals and the reaction it catalyzes. So let's take a look at some names. DNA polymerase. Now that you've gone through our course, you know what DNA polymerase is. Now, the ending ASE clues us in that this is an enzyme. We know what DNA is, and we know what polymers are. Polymers are long chains of subunits. And in the case of DNA, our subunit is what? Nucleotides. So DNA polymerase is going to put together a long string of nucleotides. So DNA polymerase connects the subunits to form a new DNA molecule. Carbonic anhydrase, again ASE, we know that this is an enzyme, and this one helps turn CO2 into carbonic acid in the blood. Now, CO2 would make our blood very acidic, and so by turning the CO2 into carbonic acid, it enables our body to deal with this. So, very helpful. Alright, there's a couple of models of enzymes. The first one here that we're going to talk about is the lock and key model. And the lock and key model is just what it sounds like. You have your substrate, which is what you start out with. This is the reactant of some kind of the chemical reaction. You have your enzyme, which is again is a protein, and this enzyme has an active site, and that's where the substrate is going to fit. If you think about a lock and a key, this would be the key, and this would be the lock. Other substrates, other reactants may bump into this enzyme, but unless it's the right one, it will just bounce in and bounce off, kind of like trying the key in the door until you find the one that works. You can put lots of keys in there, but only one of them will actually open the door. Let me go back. The other model of enzymes is called the induced fit model, and I don't have a slide on it here, but this is where the enzyme actually changes shape as the substrate reaches the activation site. This is a little more accurate as to how most enzymes function. And what happens is there's some chemical changes that occur as the substrate bonds to the active site, and it actually causes the protein, the enzyme, to change shape. And that's called the induced fit model. Much like if you see those little stretchy gloves on sale at Target and they look really tiny and you put your hands in them and they stretch out to fit. Your hand being the substrate, which doesn't change, and then the glove being the enzyme, which molds itself to your hand. So there are two types of enzyme reactions, or two ways that an enzyme can facilitate or help a reaction. There are synthesis reactions, in which synthesis means to put things together. So you have a couple of substrates, two or more, and the enzyme helps squish them all together to form one product. Decomposition means to break things down. An enzyme can also take one reactant and split it into multiple products. So that would be a decomposition reaction. We did a lab with pineapple juice. I'm not going to go over that now, except to say The effects of temperature on enzymes. When you cool an enzyme down, it's likely to not work as well. When you heat an enzyme up, the efficiency of the enzyme will decrease until you reach a certain point, and then the enzyme will be denatured. It will be broken down. And we'll talk more about that in a minute. So heating the enzymes breaks them down. Also, freezing them will slow them down, but it doesn't necessarily destroy them. The effects of pH. All enzymes have an optimum pH range. Generally, mildly acidic solutions are best. Bases will generally denature them or break them down. Very strong acids may break them down as well. So temperature and pH are the biggest regulators of enzyme activity. Our body is full of enzymes that need to have an optimum temperature that is very close to our body temperature. Imagine that. That's great, huh? So having an optimum temperature enables the enzyme to work with maximum efficiency. Each enzyme has an optimum pH also. So we've been talking about protein denaturing or denaturing the enzyme. If this is what a normal functional protein looks like, it's full of twists and curls.